And we're going to go here to share screen. Okay. All right, so let's start out with just a basic Punnett square. So we call these monohybrid Punnett squares. So essentially we're just looking at one trait at a time. So we're going to call these monohybrid Punnett squares, or you may also see them called Mendelian Punnett squares, since we're just looking at one trait at a time. So some of the monohybrid traits that we'll look at: tongue rolling, rolling is dominant, good ability to taste PTC, and in this case, tasting is dominant. Also, widow's peak, that's a Mendelian trait. Peak is dominant. Hitchhiker's thumb. Hitchhiker's thumb is recessive. I think if there was any others. Oh yes, cleft chin. Cleft is dominant. Okay, so let's say we're going to do a monohybrid Punnett square, and let's say we're talking about tongue rolling. So let's say that the parents are both heterozygous. So thinking back to our terminology, heterozygous, hetero means different, zygous means zygote. Remember, the zygote is the combination of the genes from mom's egg combined with the genes from dad's sperm cell. So if both mom and dad are heterozygous at the locus, at the location on the genes that determines whether or not you can roll your tongue, if they're both heterozygous, then we're going to represent dad with one capital R and also one lowercase r. And just so you know, it actually doesn't matter if you put dad along the top or along the side. You just need one parent along the top and one parent along the side here. And if mom is also heterozygous, then she needs one capital R to represent the dominant allele and one lowercase r to represent the recessive. So when you're doing a basic monohybrid or Mendelian Punnett square like this, each box is representing essentially 25% of generation one. So this is 25% here, this is 25% here, 25%, and 25%. So in that first little box, you're going to take, and these letters are representing the alleles, and then the combination is representing the entire genotype of mom and dad. So we're taking one allele from here from dad, one allele from here from mom. So in this little box, we would need two capital R's. So essentially what that means so far is that every single time this mom and this dad have a child, they have a 25% chance of producing a child that is homozygous dominant. Because remember, two capital letters is always homozygous dominant. Okay, and then here in this little box, so we're always putting the capital letter first, always the dominant allele first. So we're taking one allele from here, and then one allele from here. So this is going to be a heterozygous child here. And then in this box, we're going to take one allele from here, one allele from here. So we're getting another heterozygous child here. Okay, and then in our last box, we're going to take one allele from here and one allele from here. So in this last box, we end up with a homozygous recessive child. Oops, sorry about that. Homozygous recessive. Okay, so once you have your monohybrid Punnett square created, you can now essentially predict the probability that the parent will have, the child will have these various genotypes and phenotypes. So essentially every single time this mom and this dad produce a child, at least when we're looking at tongue rolling, they have a 25% chance of having a homozygous dominant child. They have a 50% chance of having a heterozygous child, since it's this box and this box. So they have a 50% chance of having a child that is heterozygous for tongue rolling. And then they have a 25% chance of having a child that is homozygous recessive for tongue rolling. Okay, so these are genotypes. Whenever you see the alleles combined like that, those are genotypes. So 25% chance of homozygous dominant, 50% chance of heterozygous, and a 25% chance of homozygous recessive. You guys with me so far? Is that part making sense? Yes? Okay. So those are genotypes. So if we want to talk phenotype, remember phenotype is simply the physical expression for that trait. So in this case, we're simply looking at whether or not the child can roll their tongue. So as long as the child has at least one dominant allele, as long as there's at least one big R, then that child will be a tongue roller, since tongue rolling is the dominant trait. So essentially, for the phenotype, there's only two possibilities. You're just looking at whether they're a tongue roller or non-tongue roller. So phenotype is just tongue roller or not. So essentially, within this Punnett square here, we have this child would definitely be a tongue roller. There's at least one big R. This child would also be a tongue roller, and this child would be a tongue roller. So 25% plus 25% plus 25% gives us 75%. So essentially every single time this mom and this dad produce a child, they have a 75% chance of having a child that is a tongue roller. Let's just put having a tongue roller. And then every single time they have a child, they also have a 25% chance of having a non-tongue roller. All right, is that part making sense to everybody? Questions? Okay, let's do one more, just what we call the basic monohybrid Punnett square. So remember, monohybrid just simply means we're looking at one trait at a time. So these are the basic Mendelian traits. So let's say this time, let's say we're talking, let's do widow's peak this time. So let's say we have a dad that does not have a widow's peak. So if dad does not have a widow's peak, he has to be homozygous recessive, because widow's peak is dominant. So the only way you would not have a widow's peak is if you're homozygous recessive. So dad is going to have two lowercase w's as his genotype. And let's say that mom is heterozygous. So she does have a widow's peak and she is heterozygous. So remember, heterozygous, one dominant allele and one recessive. All right, so then we do our Punnett square. Remember, each little section, each little quadrant is going to represent 25% of generation 1. So in this first little box here, and we always do the dominant allele first, we're going to take one allele from here and then one allele from here. So in this box, we end up with a heterozygous child. And then in this box here, we're going to take one allele from here, one allele from here. So in this box, we have another heterozygous child. And then here in this box, we're going to take one allele from here, one allele from here. So in this box, we end up with a homozygous recessive child. And then here in this box, we're going to take one allele from here, one allele from here. So we end up with another homozygous recessive child. Okay, so then when we go to do our genotypes, so remember, genotypes are the genetic makeup. So we're looking at the combination of the two alleles. So out of generation 1, there's essentially a 0% chance that they would have a child that is homozygous dominant, because none of those little quadrants, none of those boxes have two dominant W's. So there's basically a 0% chance that they would have a homozygous dominant child. There is a 50% chance, 25 plus 25 is 50, so 50% chance that they would have a heterozygous child. And then also a 50% chance that they would have a homozygous recessive child. Okay, so those are the genotype probabilities. So then if we wanted to figure out the phenotype probabilities, We have to remember that widow's peak is a dominant trait. It's a Mendelian trait that is dominant. So as long as there is one dominant capital W, then that child will end up having a widow's peak. So 25% plus 25%. So essentially 50%. Every single time this mom and this dad have a child, they have a 50% chance of having a child with a widow's peak. And they also have a 50% chance of having a child that does not have a widow's peak. Okay, so when you're working through your exercises, just be careful because some of the questions are asking about the probability for the genotype, and some of the exercise questions may ask about the phenotype. So my advice is just be careful, just read it slowly and make sure that you understand exactly what the question is asking. How are we feeling about the basic monohybrids? You guys want one more example on the monohybrids, or do you want to move on to different types? Okay, so it sounds like we're good on monohybrids. Is that true? Okay, perfect. So those are the monohybrids. So next one, let's do Abo blood typing next. So before we do the Punnett Squares, let's kind of talk a little bit about Abo blood typing in humans. So every single human will have two different blood types. We have an Abo blood type, and we also have an Rh positive, Rh negative blood type. But to avoid confusion, let's talk about one at a time. And essentially, they are determined by different genes. So the Abo blood typing is on one location on the chromosome, whereas Rh positive negative is on another. So let's talk Abo blood typing. So forget about the positive and negative for a second. We're just going to talk about Abo blood typing first. So there's four possibilities for the phenotype. You can be type A, you can be type B, you can be type AB, and you can also be type O. So these are the phenotypes here. Those are your possible phenotypes. And then genotypes, we're going to list here. So with Abo blood typing, the allele that codes for blood type A and the allele that codes for blood type B are both dominant. So if you're blood type A, your genotypes, remember that's the combination of the alleles from your parents. So you could either be homozygous dominant, like that, or you could also be heterozygous. Because as long as you have at least one copy of that dominant big A, then your overall phenotype is still going to be blood type A. So for blood type A, there's two possible genotypes, either AA or AO. Same deal for type B. If you're blood type B, your genotype could either be homozygous dominant, so two capital Bs, or you could be BO, like that, because as long as there's at least one dominant allele there, you're still going to be blood type B. If you're blood type AB, that's what we call the co-dominant condition. So that means you have an A allele from one parent and a B from the other. However, they are both expressed in the phenotype. That's why they call it co-dominant, because both of those dominant alleles are expressed in both your genotype and your phenotype. So essentially, if you're AB, you have the same phenotype and genotype. And if you're blood type O, since the O allele is recessive, the only way you end up being blood type O is if you have an O allele from both parents. So an O from mom, O from dad. Okay? And as far as who can donate to whom, if you're blood type A, you can donate to other people that have blood type A. If you, for example, somebody that has blood type A attempted to donate blood to somebody with blood type B, there would be what we call an antibody antigen reaction. Essentially, your red blood cells would clot and clump. They wouldn't be able to pass through and bring oxygen to your vital organs. You would suffer from organ failure and possibly even death without medical intervention. So if you're blood type A, you can only donate to other people that have blood type A. Same deal. Or you can also donate to AB because it wouldn't cause that antibody antigen reaction. If you're type B, you can only donate to other people that have type B, or you can donate to AB. AB is what we call the universal recipient because they can receive from anybody. And then if you're blood type O, you're the universal donor because you don't have any of those anti-A, anti-B antigens on the surface of your red blood cells. So essentially, you can donate to somebody with type AB, you can donate to somebody with type A, or you can donate to somebody with type B or another person with type O. So that's what we call type O, universal recipient, or excuse me, universal donor, meaning that you can donate to anybody. Because there's no antibody antigen reaction that would cause your red blood cells to clot and clump. So you wouldn't have that if you're blood type O, that you wouldn't cause that reaction in any other blood type. And then if you're blood type AB, you're universal recipient, meaning that you can receive blood from anyone. And then if you're blood type B, you can only receive from others that are type B or type O. And same deal with blood type A. If you're blood type A, you can only receive from others that have type A or type O. Okay, so when you're doing the Punnett squares, once you understand this part of it, so once you understand that there's 4 possible phenotypes, you can be type A, type B, type AB, or type O, and there's essentially 6 possible genotypes. Once you understand that, the rest of it's basically the same idea. So let's say, for example, we have a dad. Let's say dad is blood type AB. So we're still, same idea, we're still representing the alleles in the various quadrants of the Punnett squares. So we're going to say that dad is AB. And we're going to say that mom is blood type O. So the only way that mom can be blood type O, she needs 2 copies of that recessive O allele. So still same idea. Each quadrant essentially is representing 25% of generation 1. Okay, so in this first little box, and we always put the dominant allele first. So in this little box, we take 1 from here, we take 1 from here. So this child would be AO. And then here in this little quadrant, we take 1 from here, and we take 1 from here. So this child would be BO. And then here in this little box, we're going to take 1 from here and 1 from here. So this child would be AO. And then in this box, we're going to take 1 from here and 1 from here. So this child would be BO. Okay, so essentially every single time this mom and this dad produce a child, they have a 50% chance of having a child that is AO, but essentially overall blood type is type A. And they have a 50% chance of having a child that is BO, but essentially overall phenotype is type B. Okay, so it is not possible for this mom and this dad to produce a child that is AB. It is also not possible for this mom and dad to produce a child that is type O. So essentially 0% chance, 0% chance of having a child that is AB, and also a 0% chance of having a child that is O. All right, you guys with me so far? Questions? You guys are good? Okay, let's do one more with ABO. So let's say in this circumstance, let's say that, here I'll give you guys my parents' genotypes here on this one. So for my parents, my dad is AO and my mom is BO. But I am actually type O, and I will show you how that's possible. Okay, so here we have it set up. So we've got dad's genotype up here, dad is AO, and we've got mom's genotype here, mom is BO. So again, same idea, each little quadrant is going to represent 25% of generation 1. So here in this first box, we're going to take 1 allele from here, 1 allele from here. So there's a 25% chance of having a child that's blood type AB. And then here in this quadrant, remember we always do the dominant allele first. So 1 from here, 1 from here. So 25% chance of having a child that's BO. And then here in this quadrant, taking the dominant allele first, 1 from here, 1 from here. So 25% chance of having a child that's AO. And then here in this quadrant, They're both recessive alleles, so it doesn't matter which one we do first. We're going to take one from here and one from here. So here's me. There's 25% chance of having a child that's blood type O. So essentially every single time these two parents, every single time an AO and a BO parent have a child, they have a 25% chance of having a child that is type A. They have a 25% chance of having a child that is type B, 25% chance of having a child that is type AB. That's my sister, actually. My sister is AB. And then there's a 25% chance of having a child that is type O. So essentially for my parents, every single time, well, they're past their childbearing years now, of course, but every single time they have a child, they essentially have an equal chance of having each of the 4 blood types. They have a 25% chance of a child with type A, 25% chance of a child with type B, 25% chance of a child with type AB, and 25% chance of a child that has type O. So essentially they have an equal likelihood or equal probability of each of the 4 possible phenotypes and genotypes for blood typing. Okay. Are you guys with me on the blood typing? Do we want another example, or are we ready to move on to the next type? You guys are feeling good on this one? Awesome. Okay. All right. So I mentioned that each of us have 2 blood types. We have an ABO blood type, and we also have an RH positive, RH negative blood type. And these blood types are determined by different loci. So remember, loci, or the locus, is the location of a gene on the chromosome. So there is a locus on your chromosomes that will determine whether you're blood type AB, or O. And there's also a different locus that determines whether you're RH positive or RH negative. So for this one, this one is actually a basic monohybrid. So if you're RH positive, that's the dominant phenotype. So if you're RH positive, and they just chose a letter, they chose D's to represent RH positive, RH negative. So if you're RH positive, you're either homozygous dominant like that, or you could be heterozygous. Because remember, with the basic monohybrids, as long as you have one copy of that dominant uppercase D allele, then you're going to have the dominant phenotype, which in this case is RH positive. Okay, and then the only other possibility is RH negative. So if you have RH negative blood, that's the recessive phenotype. So the only way you could have the recessive phenotype, you would have to be homozygous recessive. You would need a recessive allele from both mom and dad. Okay, so let's say, and just like the other ones, once we understand the basics, then the rest of it, the basic Punnett Square is the same idea. So we put one parent along the top, one along the side. So in this case, let's say that dad is homozygous dominant. So we're going to represent that with 2 uppercase D's. And let's say that she has RH negative blood. So if mom is RH negative, she has to be homozygous recessive. So she needs 2 copies of the lowercase r there. So again, same idea as any other Punnett Square. Each quadrant is representing 25% of generation 1. So here, oops, I'm sorry guys, I accidentally did R's there instead of D's. I apologize. Let's fix that. Okay, so here in this first little box, and remember we always do the dominant allele first, so we're going to take one from here, one from here. So here in this box we have a heterozygous child. And then here in this box we're going to take one from here and one from here. So we have another heterozygous child. And then here in this box we're going to take one from here and one from here. So we have another heterozygous child. And then here in this box, we're going to take one from here and one from here. So we end up with another heterozygous child. So in this particular circumstance, these 2 parents, a homozygous dominant dad having a child with a homozygous recessive mother, that means 100% of their offspring are going to be heterozygous as the genotype, so 100% heterozygous. And it also means that 100% of their offspring will be Rh positive. Because remember, with a basic monohybrid Punnett square like this, as long as there's one dominant allele, then that child will display the dominant phenotype, which in this case is Rh positive blood. So every single time this mom and this dad produce a child, 100% of their offspring will be heterozygous, and 100% of their offspring will have Rh positive blood. Is this making sense? Yes? Okay, awesome. Okay, now the last type of Punnett square I'm going to show you guys is generally the hardest one. I don't say that to scare you guys, but just pay close attention because this one does get a little bit trickier. Not much trickier, but just a little bit trickier. So we're going to call these sex-linked Punnett squares. So thinking back to what we learned last week in chapter 2, in humans, we have 46 chromosomes or 23 pairs. So pair number 1 through 22, we call those autosomes. We call those essentially body chromosomes. So that's chromosome pairs number 1 through 22. And then your 23rd pair of chromosomes is going to determine your biological sex. Now, of course, there are variations, even biologically. There are syndromes such as XXY syndrome, XO syndrome. But just to avoid confusion, let's just talk about the typical condition. So typically a female is going to be XX and a male is going to be XY. So when we say the term sex-linked, all we mean is the gene and the alleles that code for that trait are located on the sex chromosomes. So if it's an X-linked trait, then it means that that gene is located on the X chromosome. So let's use an example. Let's do, actually, instead of color blindness, let's do the cat example, since that's the example in your lab manual. So with cats, the only way they end up with that calico color, and only females can be calico, or tortoiseshell. I think they call it tortoiseshell in your lab manual. So let's call it tortoiseshell since that's what your lab manual calls it. And they use bees to represent it. So sorry guys, I'm just deleting here the colorblind example. And we can do the colorblind example too, but let's do the cat one first since that's what's in your lab manual. So let's do the female possibilities. So if a female cat, and notice how I'm including the sex chromosomes here, and you'll understand why here in a moment, but just know if it's a sex-linked Punnett square, you have to include the sex chromosomes, and then you put the letter representing the allele right next to the chromosome, okay? So if it's an X-linked, it means that that allele is located on the X chromosome. So for a female cat, if she has a dominant uppercase B allele on both of her X chromosomes, and this is just a regular old black cat. Maybe I shouldn't say regular old. Black cats are not regular, they're special as well. But if a cat has an uppercase B on one X and then a lowercase b on the other, that's how you get the tortoiseshell color. Because it's incomplete dominance essentially. So the B allele, the dominant allele, is coding for the dark melanin color in the coat. And then the B allele, the lowercase, is coding for the orange color. So this is what we call incomplete dominance because they're both going to be expressed in that cat's fur. So that's what's making the tortoiseshell, or you may also hear it called calico. And then if a female cat has a lowercase, a recessive B allele on both of her X chromosomes, then she is orange. Okay, so the black coloration is dominant, the orange coloration is recessive, but if she has a copy of both, then she will have a combination. So she will be tortoiseshell, or you may hear it called calico. So for males, because they only have one X allele, they only have two possibilities. A male cat cannot be calico. If a male cat has a B, an uppercase B dominant allele, this will be a black cat. And if the male has a recessive lowercase b, this will be an orange cat. Okay, so the females, at least for an X-linked trait, females will have 3 possibilities, and males will only have 2 possibilities. You guys with me so far? Yes? Okay, so once we understand that, the Punnett squares are basically the same, or the same idea at least, but we do have to include the sex chromosomes, and you'll see why here in a second. So let's say, okay, let's say that dad is black in coloration. So on his X allele, that means he has to have the capital uppercase B allele, and he's got his Y over here. And let's say that mom is calico. So if mom is calico, then she has one dominant B allele on the first X, and then she has a recessive lowercase b on the second X. So we want to calculate the probabilities of their offspring. So remember, cats sometimes have litters. So this makes even more sense when we're talking about organisms that have litters, because you're basically calculating the probability. So here in this first little quadrant, so we're taking one from here, one from here. So this would be X big B, X big B. And then here in this quadrant, we're taking one from here and one from here. So we're always putting the X chromosome first. So this is an X big B and a Y. And then here in this little quadrant, we're taking one from here and one from here. So we're putting the dominant allele first. So this one's an X big B, X little b. So that's a calico or tortoiseshell. This one up here is black. This one up here is black. And then here in this quadrant, we're taking one from here and we're taking one from here. So in this quadrant, we have an X little b and a Y. So this is an orange male. This one's a black male. This one's a black female. Okay, so some of the sex-linked Punnett squares are going to ask you possibly about the probability for just the male offspring or just the females. So just be careful when you're doing the sex linked. If it's asking you about just the females, for example, then you're essentially only looking at the individuals or the quadrants in the Punnett square that have two X's. So you're just looking at this one and this one. So essentially every single time these two cats have offspring, a female offspring, they have a 50% chance of having a black female, and they have a 50% chance of having a tortoiseshell female. Now, if you're talking about just the males, if you're looking at just the male offspring, we're only looking at those offspring that have an X and a Y chromosome. So every single time this mom and this dad have a male or a son, they have a 50% chance of having a black male, and they have a 50% chance of having an orange male. Okay? But if it's asking about just the probabilities in general, then you could say essentially every single time these two cats have kittens, they have a 25% chance of having a black female. They have a 25% chance of having a black male. They have a 25% chance of having a tortoiseshell female. And they have a 25% chance of having an orange male. Okay? All right, let's do one more example with the sex link, because these can be a little bit trickier. So let's say this time, let's say we're talking about color blindness. So we're moving away from the cat example, and we're talking about color blindness in humans. So color blindness is an X-linked trait. It is a trait that is actually more common in males than it is in females, and I'll show you why here in a moment. But essentially, what we need to know is that that allele is located on the X chromosome, and we're going to use C's to represent color blindness or normal vision. And this is color blindness. And it's a recessive condition. So for females, they're still going to have three possibilities. So remember, females have two X chromosomes. So if there is a dominant uppercase C on both of her X chromosomes, and this is a normal vision female. The second possibility is she has a dominant C on her first X and a recessive C on the second. If it looks like that, then this is a normal vision female. However, she is a carrier for color blindness. So that means that she has normal vision herself. She can see perfectly fine. She can see all colors, but she may be more likely to pass it along to her sons. So may pass along to sons. Okay, so then the third possibility for females, the only way a female is colorblind, she would need the recessive lowercase c on both of her X chromosomes. So she is X little c, then this is a female that is colorblind. So it's not impossible for females to be colorblind, it's just less likely because they would need to inherit, that female would need to inherit the recessive C allele from both her mother and her father, essentially. So it's not impossible, it's just less likely. All right, and then for males, they only have two possibilities. So if they're X big CY, that's a normal vision male. Oops, sorry, my Microsoft Word keeps being funky. So this is a normal vision male. And then if he has a recessive lowercase c on his X, then that is a colorblind male. So for males, since they only have one X chromosome, that means if they carry that recessive allele, that recessive little c on their one X, then that male is colorblind. Okay, so once we understand the basics, the idea of the Punnett square is the same. Oops, so over here. Okay, so we have our Punnett square. So dad, let's say that dad is normal vision. So if dad's normal vision, that means he has a big C on his X and he's got a Y because he's a male. And let's say mom, let's say she has normal color vision, but she's a carrier. So on her first X, she has a big C, but on her second X, she's going to have a recessive little c. So again, same idea as any other Punnett square, every little quadrant, each little quadrant represents 25% of generation 1. So here in this first box, we're going to take one from here, and we're going to take one from here. So in this first box, we have an X big C, and then here in this box, we're going to take one from here and one from here. So we've got an X big C and a Y. And then here in this quadrant, we're going to take one from here. So we're just always doing the dominant allele first. So the X big C, then followed by the X little c. So we've got an X big C, X little c. And then here in this quadrant, we've got one from here and one from here, listing the X chromosome first. So in this quadrant, we've got an X little c and a Y. Okay, so if the question is asking you, what is the likelihood or what is the probability that these two parents would have a daughter that is colorblind? So first of all, in order to be a daughter, we would need two X chromosomes. So essentially, we're just looking at this quadrant and this quadrant. So they have a 0% chance of having a daughter that is colorblind. Every single time they have a child, if it's a daughter, they know for certain that that daughter will not be colorblind. So they have a 0% chance of having a daughter that is colorblind. However, 50% chance of having a daughter that is a carrier for colorblindness. So again, we're just looking at the daughters right now. And then as far as the sons go, so in order for it to be a son, there needs to be an X and a Y. So we're just looking at these two quadrants. So every single time these two parents have a son, so they have a 50% chance of having a son that is colorblind. So every single time they have a son, 50% chance that that son will be colorblind. Okay, right here, because we're just looking at this one and this one. So 1 out of 2, 50%. And then they also have a 50% chance of having a son that has normal color vision. Okay, so every single time this mom and this dad have a child, if it's a daughter, they can be certain that that daughter is not colorblind. However, she may be a carrier. And every single time they have a son, 50% chance that that son will be colorblind, 50% chance that that son would have normal color vision. All right, how are we feeling on the sex-linked Punnett squares? Do we want one more example of the sex-linked? Yeah, let's do one more example with the sex-linked, because these are a little bit trickier. I know you guys can handle it though. Okay, so let's say hemophilia this time. So hemophilia is also an X-linked trait. It's a blood disorder. So it is recessive and it is X-linked. And let's use H's to represent hemophilia. Let me delete all this stuff first. So we're still talking X-linked and we're still talking sex-linked. So it just means if it's an X-linked trait, it simply means that that gene and those alleles are located on the X chromosome. So that means that females are going to have 3 possibilities and males are only going to have 2. So just like color blindness, hemophilia, it's not impossible for a female to have it, but it's more likely that a male will have hemophilia. So for the females, if they have a dominant H on both of their X chromosomes, then this is a female with normal blood production. Okay, and then if the female is heterozygous, so she has a dominant H on one X and she has a recessive H on the other. So this is a female that is a carrier for hemophilia. And then the only way a female will actually express the recessive condition of hemophilia, she would need a recessive H, lowercase h allele on both X chromosomes. So an X little h, this is a female that has hemophilia. Okay, and then since males only have one X chromosome, so remember that those alleles, those genes that code for normal blood production or for hemophilia, those are located on the X chromosome. So if there's an uppercase dominant H on that male's X chromosome, then this is a male with normal blood production. Okay, and then if that male has the recessive lowercase h on his X chromosome, then this is a male with hemophilia. Okay, you guys with me so far? So it's the same idea as the cat example, same idea as the colorblind example. We're just looking at a different trait that is also X-linked. Okay, so for this example, let's say, that dad, has hemophilia. So that means on his X chromosome, he has that lowercase h. And let's say that mom does not have hemophilia, but she is a carrier for it. So she's got a dominant H on her first X, and she's got a lowercase recessive H on her second. So once we have it set up, same basic idea. Each little quadrant is essentially representing 25% of generation 1. So here in this first quadrant, we're going to take one from here, and we're going to take one from here. So we've got an X big H, X little h. So that's a carrier female. And then here in this quadrant, we have one from here, and we have one from here. So that's an X big H and a Y. So that's a male with normal blood. Okay, and then here in this little quadrant, so we're going to take one from here, and then we're going to take one from here. So we've got an X little h, so that's a female with hemophilia. And then here in this quadrant, we're going to take one from here, and then we're going to take one from here. So we've got an X little h and a Y, so that's a male with hemophilia. So these combinations here, so they have a pretty good chance of having a child that does have hemophilia or is a carrier for it. So in this particular example, let's look at just the females first. So they essentially, if we're looking at just the females, in order to be a female, let's start there, in order to be a female, we need 2 X chromosomes. So we're essentially just looking at this half of the Punnett square, if we're talking about just the females. So essentially 50% of female offspring will be carriers for hemophilia, and 50% of female offspring will have hemophilia. Okay, and then with the males, so 50% of male offspring will have normal blood production, and 50% of male offspring will have hemophilia. Okay, so for this particular combination, there's a pretty high likelihood that they're going to have a child that either has hemophilia or is a carrier for it. So sometimes it depends upon what trait we're thinking of, but sometimes they can use this information to determine the likelihood of two parents producing a child that might have a disorder like this. Now, hemophilia isn't necessarily a death sentence. It can certainly be treated medically. But sometimes a family planner or a genetics counselor may use Punnett squares like this to help determine the likelihood of two parents having a child that has hemophilia, for example. Okay, is this starting to make sense? Yeah, okay. All right, so I did it a little bit differently today, especially since we're meeting later in the week. I assumed most of you have probably already gone over the PowerPoint by this point, so I thought it might be more worth our time to go over some Punnett squares. But do you guys have any specific questions about the PowerPoint or questions about the lab exercises that you've come across? Claire, I think you're on mute if you're trying to ask a question.
Oh, I was just saying, I don't think that I have a question. I was looking through my notes, but I think you answered them with replies to my comments. Yeah.
Oh, okay, great, perfect. All right, here I'm going to exit share screen here for a second just to make sure I don't have anything in the chat box. Okay, so I have a question here asking to explain genotype and phenotype. So the genotype is the genetic makeup. So let's go back here to our Microsoft Word document. So the genotype is the letters, essentially. So if you're looking at the basic monohybrids, this is the genetic makeup of the organism. Okay, so it's essentially the letters. So it doesn't really matter what trait you're talking about. If it's a basic monohybrid Punnett square, there's going to be three possibilities. So first possibility, homozygous dominant. So that would be two uppercase letters. For example, big A, big A. So each uppercase A is representing one of the alleles. So recall from chapter 2, we're getting 23 chromosomes from mom's ova, from her egg, and we're getting 23 chromosomes from dad's sperm. So at the locus, at the location on the chromosome that determines this particular gene, this particular trait, if there are two capital A's or two capital R's or two capital T's, whatever it might be, anytime you see two uppercase letters, you're always going to call that genotype homozygous dominant. So you break it up, first part of the word there, that means same. And then zygous, that means zygote. So recalling again from chapter 2, a zygote is a fertilized egg cell. So that's 23 chromosomes from mom's egg, 23 chromosomes from dad's sperm. So if you're homozygous dominant at a particular locus, let's say we're talking tongue rolling, for example. So if you get a dominant allele from both mom and dad, then you're a tongue roller and you're also homozygous dominant. And then the second possibility, you could also be homozygous recessive. So that's represented by two lowercase letters. So for example, if you get a recessive allele at the locus that determines tongue rolling from both mom and dad, then you are homozygous recessive and you are a non-tongue roller. And then the third possibility would be heterozygous. So one uppercase letter and one lowercase letter. Okay, so it would look like this, one big R, one little r. So if you break that word up, hetero means different or alternative, and then zygous means zygote. So that means you get a dominant allele from one parent and a recessive allele from the other. but essentially the combination of those alleles that you receive from your parents, if you have one dominant and one recessive, you are heterozygous. If you get a dominant allele from both parents, in this case ours, a big R from mom, a big R from dad, then you are homozygous dominant. If you receive a recessive allele from mom and dad, two little r's, then you are homozygous recessive. So for the basic monohybrid Punnett squares, there's only 3 possible genotypes, okay? And then phenotypes for the basic monohybrid Punnett squares, there's only 2 possibilities. So with the tongue rolling example, we're simply looking at whether or not that organism, that person, is a tongue roller, which is the dominant, versus non-tongue roller, which is recessive. Okay, so if you're the dominant phenotype, you could either be homozygous dominant like that, or you could be heterozygous like that. But if you're a non-tongue roller, your only possibility is homozygous recessive, because you would have to have that recessive allele from both mom and dad. If you have one dominant allele and one recessive allele, then that dominant allele will mask the expression of the recessive in the phenotype. Okay, so for the basic monohybrid Punnett squares, there's only 2 possible phenotypes, but there are 3 possible genotypes. Okay, so think about the genotype. It's the genetic makeup, it's the letters, and then the phenotype is simply the physical expression or the physical manifestation of the genotype. Okay, so it's essentially how that organism looks for that trait. So in this case, we're just looking at tongue rolling versus non-tongue rolling. Okay, so one more example as far as genotype versus phenotype. Let's say we're talking about widow's peak. Okay, so widow's peak is dominant. So if you get 2 dominant alleles from both of your parents at the location that determines widow's peak, then you are homozygous dominant, and you also have a widow's peak. Okay, and if you are homozygous recessive, meaning you get a recessive allele from both parents, then you are homozygous recessive at the locus that determines widow's peak, and you do not have a widow's peak. So they call that a straight hairline. And then the only other possibility as far as genotype, if you inherit a dominant allele from one of your parents and a recessive allele from the other, then you are heterozygous. But since the widow's peak is a dominant trait and there is at least one dominant allele, then this individual will have a widow's peak. Okay, and then for phenotype, remember phenotype is simply the physical expression or manifestation of that individual's genotype. So essentially, how does that person look? So we're just looking at widow's peak versus straight hairline. So widow's peak is the dominant phenotype. And then the straight hairline is the recessive phenotype. Okay, so as long as you have at least one capital W, you're going to have a widow's peak. So both the homozygous dominant individuals and the heterozygous individuals will ultimately have a widow's peak as their phenotype. And then the individuals that have a straight hairline, that's a recessive phenotype. So in order to have the straight hairline, you must be homozygous recessive. So little w, little w. Does that answer your question as far as genotype versus phenotype? Perfect. Okay. All right, do you guys have other questions about Lab 3, Chapter 3? Anything from last week that came up that's still confusing? Okay, so it is Thursday. I know we're meeting a little bit later this week than normal. But so today is Thursday, so my recommendation is try your very best to get your first attempt on Lab 3 exercises and concept review questions. Try to get those in by tonight. That way I can go through and grade it all tomorrow, provide feedback in the grade book as needed, and then you'll still have all day Saturday, all day Sunday to submit correction if you need to. And then next week, we're going to do forces of evolution, and we're actually going to do an activity that is not in your lab manual, simply because all of the activities in your lab manual for lab 4 require that you buy supplies, and I don't want you guys to have to do that. I think it's enough that you guys have paid tuition and that you've purchased your lab manual. I don't want you to have to buy anything else. So I actually have a different exercise for us to do next week that is not in your lab manual. You're still going to do your concept review questions from your lab manual, but we're going to do a different exercise. But with that being said, and I'll put this in your announcements for Monday morning, but it is really important that you come to the Zoom meeting next week, okay? Because we're doing an activity that's not related to the lab manual, okay? But other than that, do you guys have any other questions for me? You guys are good? Okay, so I will, once this recording is available, I will post it in Canvas, because I know obviously we're meeting at a slightly different time than we normally do. I understand that some of your peers may not have been able to come. So if I'll post an announcement about this as well, but if you happen to talk to anybody in your lab group, just let them know that the recording for this will hopefully be posted within a few hours. It does take, it takes me a little bit of time to transcribe it, but once I get it transcribed, I will post it for you guys in Canvas. But that's all I have for you guys today. I will see you guys on Tuesday, and I hope you have a great weekend. Oh, good. I'm glad it helped. Thank you, Sydney. Have a good weekend. Thank you, guys.